Hey, check one test. Check one test. Check. Check one test. Check. Check. Hey, check one test. Check. Check. Check one. Check one test. Check. Check. One test. Check one test. One test. Check. Check. Check one test. प्रोजेक्टर आपको वैसे गुड़गांव चाहिए था आपको मैं आपको कब चाहिए आपको 3 तारीख को एक बार मैं आपको शाम को बात करूँ हो जाएगा लगभग आपका मैं थोड़ा सा कहीं बिजी था मैम हाँ ओके मैं करता हूँ शाम को मैं ना आपको थोड़ी देर में व्हाट्सएप डालता हूँ टाइमिंग क्या रहेगी आपकी सुबह मैम अप्रॉक्सीमेटली गुड़गांव में आपको चाहिए बताइए मैं जरा से कबाड़ से भेज रहा हूँ यस मैम प्रोजेक्टर वाला Hey, check. One, test. Check one, test. Hey. Hey, check. One, test. Check. 1. Check this. Check 1. कर दिया। किसका? कहाँ का? जूम का? ये यूट्यूब का मैंने बढ़ा दिया एक बार। हाँ टेस्ट। Hey, check 1. ऑडियो ठीक आ रही है?
Now start. A very good afternoon to everyone. On behalf of everyone at IGIB, we are thrilled and we know why we gather here. We are awaiting the arrival of Professor Meldal. And meanwhile, it has been really interesting to host science of top recognition and understand the journey from their lens. So as researchers, it's an honor to host a Nobel laureate. That goes without saying, but it's also something more interesting from the point of view of interdisciplinary approaches. At IGIB, one of our mandate being genomics, we always have strengths from other disciplines, including chemistry, computational sciences, and also now more and more physics and engineering approaches are also being utilized. If you notice, one could argue that this is called as the century of biology, because most of the applications are centered around biology, but one could argue that it's mostly the periphery of biology, which is the approaches that work or come from non-biological background have seeped their ways into the core of biology and have their interesting applications. And I think that's where it really gets very interesting for today's talk as well. So I welcome all the students from DU colleges, Hindu College, Daulatram, Bhaskaracharya, Miranda, and of course the whole IGIB scientific staff and students and faculties. We are very happy to see a full Audi here. What we are now going to start the program with will be a talk about applications of click chemistry in the area of biology. And that's where mostly where my introduction was, and it will be elaborated with examples from our very own Dr. Kausik Chakraborty. So I, with no further delay, I let the stage be yours.
Good afternoon, everyone. Welcome to IGIB. Now, yeah. So it's important. So I think we are going to hear more about the journey and the discovery process from the expert himself, the discoverer himself. What I'm going to tell you is why it's important for us. The whole chemistry that was discovered, why it was important for us. So in order to understand that, just hold on. So if you look at this graph, this is the number of papers saying click chemistry over years. And you would see that there were a few papers which said that in the 60s. It also said that in the 70s. There were a few papers in the 80s. But suddenly after 2001, 2002, you start seeing this jump. That's when a new field develops. And when you see this jump, that then some breakthrough would have happened at this period, which allowed this sudden expansion of this field. And that's exactly what had happened at that point of time, which fetched 3 people Nobel Prize on click chemistry. And one of them would be here with us, and he's going to talk about one part of the click chemistry reaction, the other part being discovered by Sharpless. So what is click chemistry? I won't go into the details of the mechanism and everything, but what is click chemistry? It's basically what people were trying to figure out is, can we have 2 groups, which is C1 and C2, doesn't matter what these groups are, can we have these 2 groups inside the cell or in C2 form one moiety, like to form covalent reaction, to form one particular group through some means, so that and such that they do not react to any other molecules inside the cell, which is very difficult to imagine, because you can think about it, anything which is reactive is going to also react to things which are inside in the cell. There are multiple electrophiles and nucleophiles in the cell, so you need to design a nucleophile-electrophile combination which will react to each other and not to anything else inside the cell. So that was what the challenge was. So it took 30-40 years to get there. So the first paper on a click reaction came in 1950s, which is the top part of the reaction, which is given here. You see, it's Higgs and addition reaction, that 1,3 dipole addition reaction. The Stoffhofer reaction and the Huygens reaction was known from 50s, 60s. Yet, it took till 2001, 2002 to get here. And so once this reaction was developed, okay, so initially it was heat, and that's why you could not use it inside the cell. And now it was a copper catalyst, which we are going to hear today. Now, why is it so important? Because you have these two groups now, two very small groups. The R1 being either an alkyl group or an aryl group. So R can be anything. You know, it can be part of your protein, it can be part of your DNA, it can be part of any other small molecule. As long as you can add this azide group to one part and an alkyl group to the other part of your molecules, if they you can put them together, and they will form a covalent compound where the R1 and R2 are linked to each other. So that's the click. So it clicks and it binds these two molecules together. That's all. So this is click chemistry, and we are going to hear more about it. How do we use it? So if you look at the number of companies which sell click chemistry reagents now, it has boomed. There's a large number of companies a large portfolio of chemicals which are based on click reactions. Now, without naming any one of them, let's say, so how do we use it in the lab? Say you identify, so one of the best uses is this. So you identified a small molecule using some screen, okay? So we do screening for small molecules. Many people do that, right? So let's say from our lab, I'm giving an example. It's not that we have done it, but I'm giving an example, just an example of it. Say you are trying to figure out molecules that prevent protein aggregation or can dissolve protein aggregation. So you did a screen where you take a cell where the proteins were aggregating. It formed a fluorescent puncta, a red colored puncta. And you tested large number of small molecules, and you obtained a small molecule, which now, in the presence of which you don't find a puncta. Now the question is, no, it's a 1 million compound. It's a 1 million compound, absolutely. The reason why you cannot say, why you would not want to have hypothesis is also interesting, because there was a lot of work which was done to find out molecules that bind to aggregates and thereby which prevent aggregation. That actually turned out to be more toxic than helpful. Okay, so then people went to the unbiased approach. No, so this is not my work. So right. You are right. The question is important. Like, how do you choose small molecule? For each, you have different things. For example, you may have a chosen scaffold based on your target of interest. Let's say you did that and you found out one molecule binds, but still into FDA you have to prove that this molecule actually binds to the protein that you are talking about. And that's a challenge, because pulling down the small molecule with its binding target is a challenge still. Now, how do you do that with click chemistry? So what do you want to know? You want to know this small molecule that you obtained as hit, what does it bind to in the cell? Without that, your FDA is not going to approve the drug for any sort of trials, not even phase 1 clinical trials. So in order to do that, what you need to do, what is done generally, is that you take this C1 part, the azide part, and you add it to the small molecule synthetically of your choice. Okay, so you have this green one which attached to this C1 part, which is shown as a half moon, a crescent moon. Now in the cell, the small molecule is going to bind to its target, which I'm shown as gray protein here. Now the other thing that you need to have with you is this C2 part, which is the alkyne part, which is attached to some handle that you can pull down. Okay, some handle. For example, it can be a biotin, because biotin you can pull down with streptavidin. It could be a 10x his stack, because you can pull down his stack with cobalt or nickel column. So anything that you can, any handle, this ring would be any handle that you have a very nice hook to pull down this molecule, right? And once you incubate this in presence of copper or whatever this catalyst would be, you find a covalent reaction which takes this blue part C1 and C2 part together, and you can pull down this complex using the hook that you now have against the ring. That would pull down the protein. You can now identify the protein using mass spectrometry or other means that if it's a hypothesis-driven research, you can do a Western blot to figure out whether it binds or not. If it's a hypothesis-free research, where you do not know where it's binding to, you can do a mass spec, exploratory mass spec or discovery mass spec to figure out what the protein is that's binding to your hit, which is important for you. And then you can do all the biophysics down the line. So this is where it has become very important, from hit to identification of targets. Okay, and I'll give you one more example that Sheetal pointed out to me. Thank you, Sheetal, for pointing this out to me. This is a very interesting way that it's used. So in cancer, apparently, serine proteases are big targets. But we do not know which serine protease is active in cancer cells. So how will you figure out which serine protease is active in a particular cancer that you're interested in? So the way it is done is this. So let's say that, this is the gray one here. It's the same concept that I showed last time, a little, just a variant of the same concept. So you have this gray one, which is the serine protease. What you would use here is a suicidal inhibitor of serine proteases, which are generally available. Okay. So what is a suicide inhibitor? Suicide inhibitor is a molecule that would act as a substrate of the serine protease. And if the serine protease is active, it's going to act on the substrate. The substrate is going to covalently attach to the serine protease and not leave it. And it's going to kill the enzyme. The enzyme activity is single turnover. It reacts with the substrate, the suicidal substrate, makes a covalent bond with it and doesn't let it go, and then the enzyme becomes inactive. But inhibition is not you are not trying to inhibit the enzyme. You are trying to figure out what enzymes are active as serine proteases inside the cell. You have to understand, serine proteases are kept inactive by a lot of propeptidases and its binding partners. So if you just do a mass spec, you will find a lot of serine proteases, but you will not know which of them are active serine proteases inside the cell. Okay. So if you want to figure out the active serine proteases. So what will happen? So what you do here is, you take a suicide inhibitor and you add a C1 handle to it, or an azide handle to it, or an alkyne handle to it, and then you add it to the cell. So what would happen is the active serine proteases are going to get covalently linked to the green one, right, the suicide inhibitor, which now will put the C1 handle to the serine protease. You can now again use the same trick as we showed earlier, that you take the complementary C2 molecule, which is bound to a ring being your handle of pulling down, and then you can pull down the serine protease and figure out which serine protease is active in the cell. So this gives you an idea about how to use small molecule C1, C2 type condensation reaction. You need copper for this reaction, but for the other Nobel laureates' work, you don't need a copper. You need, so originally, in order to facilitate this reaction, you needed heat. So one group figured out that you can use copper to do the same reaction. Another group figured out that you can create a alkyne which is heavily strained by a ring. And that strain allows it to be more reactive, and thereby it reacts faster. So there are two types of C2. The azide being the same, C2 is different in these two cases. That's called bioorthogonal click chemistry. This is the copper catalyzed click chemistry. Okay, so, but idea is same. The idea is that you would require copper, and in this case, you would require copper. In the other case, you don't require it, and you can do the click chemistry reactions. And so the way we use it in the lab is to figure out newly synthesized proteins, which is a very interesting way of using click chemistry. Now, if any one of you have done measurement of translation rate, you would know that it's typically done by using radioactive methionine. So the way it is done is this, that you take away methionine from the medium. From the time that you want to measure translation, you add radioactive methionine. So the newly synthesized proteins are going to be radioactive because it's going to have radioactive methionine in it. And thereby you can look at an autoradiography and figure out, autoradiography would allow you to figure out that with how much protein has incorporated methionine. But that's one, it requires a radioactive setup, which is not there everywhere. And secondly, it requires a lot of safety measures, which is not so easy to have in all different setups. So there was an important reason to measure translation. One is to measure translation, the other is to also figure out newly synthesized proteins. So you give a stress to a cell or something to a cell, and you would want to figure out immediately what are the proteins that are being made at that point of time. So how do you label only like newly synthesized proteins. The way to do it is metabolically by either using some C14 label leucine kind of methods. But this is a very interesting method, and this is the most suitable method now. So this was started. This started off with this particular molecule that we are going to talk about. Now, what do you need? You need to label the protein with either C1 or C2. In order to have a handle to pull it down, you would have to label the protein with either C1 or C2. So you need to incorporate an azide group into the newly synthesized proteins or an alkyne group. Proteins don't have azide group or alkyne group, and obviously they don't have, otherwise click chemistry will not work. Because if you had put in an alkyne, otherwise they would have reacted to the endogenous azide. So these two groups are not there. So now the question is, how do we put that in? So this was a paper in 2002. It's a seminal paper again, a very seminal paper. In 2002, which figured out that this particular molecule, which is called azide-alkyne, can anyone tell me which amino acid does it look like? Which amino acid does it look like most? At least in terms of the number of atoms or the side chain. No? No, glycine doesn't have a side chain, no? So this looks most like methionine. Okay, so if you look at the charge, and so they modeled the whole thing, this has all the properties of methionine. So methionine would end here, actually, SH. But they figured out by experiment that this can be easily taken in by the cell, charged on the methionine tRNA and incorporated in proteins without any problem. That was amazing. Like now, if you add this to the cell, instead of methionine and take away methionine from the media, this will get incorporated at all the places that methionine would be, and it will level most of the proteins, because most of the proteins do have at least one methionine. Most of the proteins, not all. Okay. Now, important. Why did I put this title? I put this title to highlight two things. It was published in 2001. This was before the original Nobel Prize winning click chemistry was published. A month before or so, because the EPUB was 2001 December. This was around the same time it was discovered. So this was not meant for the click chemistry Nobel reaction that we know today. It was meant for the Staudinger ligation. The Staudinger ligation is an earlier version which required heat and other things. So these were all for in situ. You have to isolate the molecules, you have to do a heating and the reactions for that. So this molecule was made before the click chemistry.
So these people arrived at this by all permutation combinations or from third
No, this was completely hypothesis driven. So they modeled methionine. They had to have an azide. So they made 4 different variants where they had azide at this place or this place and at this place. So it was completely screening plus hypothesis driven work. So if you put this, now your protein has a C1 group on it. Great. So if you have a C1 group, then the job is done. As long as your protein the tRNA gets amino acylated with the methionine tRNA, and then the methionine tRNA gets amino acylated with this particular one, and then your protein has a azide group. Now it's simple. Now the next steps are as simple as this. Now you have a newly synthesized protein. You have the C1 group to it, and you can pull it down. Simple. And you can identify which proteins are made at a time t is equal to 0 after you've added this. Absolutely.
So this protein visualization is as sensitive as audio radiography?
Okay, so the question is whether the protein visualization is as sensitive as audio radiography. Yes, it is, because the visualization done using this handle is actually fluorescence. So you're not pulling it down. For visualization, what you do is you have the C2 attached to a very sensitive fluorophore. So you can then just run it on the gel and quantify the fluorescence. You can directly quantify the fluorescence in the cell using flow cytometry. So you have an in vivo measurement of translation rate. Okay, so this is generally sold as a kit now. So it's an in vivo measurement of translation rate. The kit is available with multiple companies. We have used one of them and it works fine. So this is in vivo click chemistry. You can pull it down, do mass spectrometry to figure out newly synthesized proteins. So you can do all this sort of stuff. For example, if you are, our lab works on protein folding. So you can, the obvious reason would be to figure out what are the chaperones that interact with newly synthesized proteins. You can think of this way. So you can, when you pull down the newly synthesized proteins, if you pull it down in the right time, they will be associated with chaperones. And you can pull down the chaperones and you can identify which chaperones are being, which chaperones are interacting specifically with when newly synthesized proteins. So these are sort of, tools. So you can label proteins, you can label small molecules. For nucleic acid, we'll get to, for nucleic acid and first-hand experience, we'll get to the people who have done that. So Shobik and Debo have done some work on first-hand on this click chemistry. So I'll ask Shobik to introduce that work to us.
Yeah, schedule is as follows that, we are still waiting for a message because before our, schedule there is a engagement of Nobel laureate to meet with minister. So still that meeting has not started yet. Okay, and we have checked in Google, it will take almost 50 minutes to come from headquarters to our institute. So we are planning to have a small break in between. Then again we will come back here to listen the talk by Nobel laureate. So here what I am going to do is that, what we have used in our lab, the advantage of this click chemistry. This is the paper which appeared in 2009 in Langmuir, and we have collaborated, 3 scientists, PI has collaborated for 3 different reasons. The concept is that dendritic effect. What is dendritic effect? To give a simple example is that when we talk about, say, drug, okay, is binding to a protein, how much it will bind, it depends on the local concentration. It doesn't depend how much you are treating. How, say, if the protein is within a cell and you are targeting to the cell, then how many ligand molecule is entering, okay, to this particular cell, that will decide the efficacy of that ligand. Okay. So local concentration is the concept. Now if this ligand somehow you are making artificially concentrated and sending to a particular cell, the local concentration of that ligand will go up, so that efficacy will increase. That's in other way we call dendritic effect. So how do we have dendritic effect for a small molecule, so that they all together go in a particular place and increase the local concentration, so that efficacy will be higher. So what we have done, 3 of us, me, Prashad and Hota, 3 of us are close friends. Both of them, Prashad and Hota, belongs to, that time he used to belongs to NCL, National Chemical Laboratory, and I used to be in IGIB, and IGIB has a small lab in NCL. So 3 of us, we decided to couple ourselves, our own expertise to address this issue. So Prashad synthesized nanoparticle, and Hota modified that CO1 and C2, which basically Kausik explained, and rest of the work I have done. The synthetic scheme is like that, as follows. So here, this is the therapeutic molecules, barbarin. It is known as a anti-apoptotic molecule. Okay. Now if we increase the local concentration of this molecule, the efficacy will increase. So we modified this molecule, attached that C1, N3 azide group into 3 steps. First, this molecule basically is planar molecule. It binds to the protein pocket, just like an intercalation what happens in case of DNA. And this is the part basically goes into the pocket of protein, because as you understand there is no modification, completely planar. This part of the molecule is also planar, but due to these 2 side chain, the steric hindrance to penetrate in the planar space is difficult. So we took advantage of this and we modified this portion, and 3-step reaction. With this, we have added the azide group, the N3, which is one of the molecule functional group which you need for click chemistry. Now, in other way, Prasad, what he has done, he has taken a nanoparticle, synthesized. That time, buying nanoparticles was not available in the commercial, so from the commercial source, so he synthesized this nanoparticle in his lab, which is very monodispersed, again modified and introduced the carbene group, the C2 group which you require for click chemistry. Now you mix these two molecules or two systems in presence of copper, they click, which Kausik has described, and as a result the surface of these nanoparticles get coated with berberine. Now depending on the surface area, you can decide what should be the local concentration of this molecule. So you can vary also. So that's the experiment we have done. We have not only varied the concentration of berberine molecule around the nanoparticle and changed the dendritic effect gradually, but also we have taken advantage of the linker length, how far it has to be from the surface of the nanoparticle, so that it retains its activity. So that's the work we have done and shown that dendritic effect can be captured on nanoparticles to enhance the efficacy of any kind of therapeutics molecule, and click chemistry is one of the important reactions here to take that advantage. So this is the first example which we published in 2009.
How are these nanoparticles targeted to cells, specific cells?
So in this paper we have not addressed that issue, but subsequently we tried, but we could not follow this work. We used to use kind of ligand for a receptor for the cell surface. So it is a mix of berberine modification as well as a receptor in a certain ratio, say 9:1, and then we put in the reaction mixer, expecting that out of 10 molecules, 1 molecule will be ligand for a particular receptor. And the chain length also used to vary such a way that initially it will be exposed. Like if you keep chain length of the ligand specific for the receptor is little bit higher than the berberine chain length. So initially cell surface will see the molecule, ligand molecule which is targeted for the receptor. So it will be taken within the cell. So that's what we have tried, but we could not publish that paper. It was very complicated to show that that time that it is really taken up with a particular cells but not by other cells. So we could not, we are not that mature biologists at that time, and we could not, but we tried that one to make it targeted therapy. So in second, this is a very recent example. Again, 3 of us we collaborated, myself, biophysical chemist, Devu, a CRISPR-Cas biologist, and Srivastan from ISAR Pune, who basically is synthetic chemical biology for nucleic acid. So as those who are from IGIB, we know Devu is a very actively working on CRISPR-Cas technology, and the advantage of CRISPR-Cas technology is that you can target or send this complex molecule, RNP complex, at any part of genome, whatever you are designing, wherever you are targeting. Okay, and if you silence the enzymatic activity of the Cas9 protein, it will not be able to cleave, but it will be able to bind in a particular place or particular site of the chromosome, wherever you want to design. That is the designing of the sgRNA. So that means it can target particular location of genome. That's the CRISPR-Cas technology's advantage. Now if I want to visualize it, okay, where it is going, then what is the microenvironment, then I have to take the advantage of fluorescence microscope. Okay, fluorescence does not only tell you the location of the molecule, but also give different kind of microenvironmental information like polarity, like pH, like viscosity, that you can measure just measuring fluorescence intensity of different kind of probe. So there is applications. So what we did, basically Srivastan has synthesized modified nucleotide base, which is shown here, and it can be incorporated enzymatically at the end of any RNA molecules. Okay. During that, he is introducing one carbene molecule, so that when it is getting extended with this unnatural base, that all the bases is getting labeled with a functional group which is basically liable for click chemistry. Now use this molecule. Yes, so that enzyme's name, uridyl transferase we have used. Okay. So now, now use this system to synthesize basically a sgRNA which is getting extended with some modified base, chemically synthesized base, and one of the side of this base is having that C2 molecule which is needed for click chemistry. Now once you have this system, you can attach anything you want, any kind of fluorescence probe molecule. It could be something else, it could be even drug molecule. Say you want to deliver a particular drug in a particular location of a chromosome to change some epigenetic modification you want to bring in. Okay. You can introduce that kind of molecule and target it using CRISPR-Cas9 advantage, and you can make a kind of chemistry or chemical change in a particular location of the genome. That's the way we have did it. We basically used in this case for as a proof of concept, used fluorescence moieties, and showed that indeed it is possible to label sgRNA by click chemistry that can bind to Cas9 protein without losing its efficacy of binding. Then the RNP complex can go, now it is modified, kind of chemically modified system. It may not act like a natural. So we have proved that even though modification is there, they retain their all kind of parameters, like they bind in a same location of chromosome and go and sits there. Now we can use these N3 molecules or azide molecule and put any kind of carbene molecule, attached molecules including fluorescence molecule. It can be labeled, you can visualize, you can measure microenvironments like polarity, pH, and so on and so forth. That's what we proved in this paper, and eventually we published all together in JACS.
So since it can take care of off-target issue,
CRISPR-Cas can take care of target issue. Yeah.
are people using for
So very recent, work. This we published in 2020, I believe, or 21. Eventually, people may adopt it, because this is kind of first example using click chemistry as well as Cas9 technology advantage to kind of understand different location of chromosomes. Okay, so that got published in JACS last year. With this, I will end here. This is the two examples which we have done ourselves in this institute. Is there anything else? Okay, so I will stop here. I will hand over podium to Lipi to continue.
Thank you so much, Kausik and Shavik. So you can see why there is an enthusiasm in IGIB. We have a director whose background matches completely with the incoming guest. What I'm going to now shuffle the program a little bit. We had the high tea towards the end of the program for the students. I'll request now for the students to go to the lawns for the tea and be here in half an hour. Okay, by that time he'll arrive and we'll also utilize the time. So please join us at the lawn for the tea. Thank you.
Please don't eat inside the auditorium. Those who have food boxes, please go out and eat. Then come back.
Check one, test. Check one, test.
Hello, everyone. Just an update for the audience. Professor Meldal will join us in 10 to 15 minutes. And we'll start immediately now. I just have a request that we will be doing a photo session with him after the concluding remarks. So there will be a photo session on stage with faculties of IGIB and DU college teachers. And then they will exit from the gate there. Because we are full on both the sides, I would then ask the students to come on the stage, but please don't create any rush. There is enough place here to have photograph for the students as well. Okay, so some of you, if you can just sit on the on the floor here, we'll try to accommodate everyone. Okay, I hope that's okay. That's the best we can do to have a group photo session. Okay. And yeah, and we are back to the waiting mode. Thank you.
Hello everyone. Please settle down. In 2 to 5 minutes, he'll be here. Request everyone to silence your phones, please.
So good afternoon, everybody. So first of all, apologize because of late, because professor was having a meeting with minister. So as you know that cannot be scheduled in time. So without much introduction, let me join with me to welcome Professor Meldal and his wife. So as this is a very small institute, we are 40 scientists, almost 400 research workers. We are working here. We are the part of a larger chain of institute known as Council of Scientific and Industrial Research. Just now we have visited the headquarters. So our institute basically is dedicated for genomics research in the country. We are the institute who has basically given the first draft genome for the country, as well as we also involved at this moment a large scale population genomics study. Apart from genomics research, our institute also are involved different fundamental biological research related to human health. Without much introduction, I will invite my colleague Lipi to introduce today's speaker, for which we are waiting for a long. Thank you very much.
Thank you, Shovik. I am Lipi Thukral, a computational biologist at this institute. It goes without saying that all of us, as scientists, as researchers, are just thrilled to host Professor Meldal here. I'll just brief you. We are joined here by PhD students, research staff students, faculty at IGIB, and in addition, we have students from Delhi University, from Hindu College, Daulat Ram, Bhaskaracharya, Miranda House, and yeah, we are all just very much looking forward to your talk. But previous to that, I just want to introduce the speaker who definitely needs no introduction, but as you all know, we have just been briefed about his work on click chemistry by our faculties, Dr. Kausik Chakraborty, and also our director, Dr. Kausik Methi. Professor Meldal was born in 1954 in Copenhagen in Denmark. And he was awarded a MSc degree from the Faculty of Chemical Engineering at the Technical University of Denmark, and later also graduated from DTU in 1983. His PhD supervisor was Dr. Klaus Bock, and they worked together in the area of carbohydrate synthesis, structure, and immunology. And he's shared this, that his PhD supervisor was quite demanding. We'll come to that later. After a 2-year postdoc at DTU, he completed, which he completed at Laboratory of Molecular Biology at the University of Cambridge. He worked with Professor Robert Shepherd. Immediately after the postdoc, he returned to Denmark. So you will see that most of his career trajectory is at its home country, which is Denmark. And in 1986, he became associate professor at the University of Copenhagen. From 1988, Meldal worked at the Carlsberg Laboratory, becoming professor in 2002. And since 2011, he's a professor at the Department of Chemistry at the University of Copenhagen. He's also the director of the Center of Evolutionary Chemical Biology and has co-founded 3 startups, and most recently Betabap in 2019. We are also glad that he's joined by his wife, Dr. Fidris and Hilaire. Thank you so much for joining us. In 2022, as we all know, he became the recipient of Nobel Prize in Chemistry along with K. Barry Sharpless and Carolyn Bertozzi for the development of click chemistry and bioorthogonal chemistry. Today, he would be speaking about molecular click adventures, a leap from the shoulder of giants. So before we all start, I just want to give a round of applause for everyone before he joins us on stage.
First of all, thank you. Maybe I should have a microphone. In this area, yeah. But I can go out there. Yeah. Thank you very much. Thank you for waiting, and excuse me for being so late. We were caught up in traffic here in Delhi. It's not always easy. We had a meeting with your Minister of Science and Technology. But thank you to CSIR for this invitation to come and present today. It will not be about genes, because that's not my specialty. It will be about molecular click adventures. That is molecules that we could not make before, which have become possible through a new kind of chemistry that was termed click chemistry. So we are very honored that click chemistry was awarded. And click chemistry is not only me, it's also Carolyn Bertozzi and Barry Sharpless, 2 good friends that shared this prize. Because click chemistry opens up a whole new box of tools that we can use in chemistry. When you get a Nobel Prize, you have the honor of actually coming also with some news about what is important here in life. And one of the things that is most important here in life in my opinion, is that chemistry is everything. And that's meant very literally. It's chemistry when you get tired, it's chemistry when you fall in love. It's chemistry when you are hungry. Your car is chemistry, your computer is chemistry. All of that is molecules that interact, that has been constructed in some way. Much of it we are actually molding every day in our production of goods. That's chemistry, all of it. So why don't we teach our kids from the very beginning, just as important as reading and writing, what chemistry is all about? Make that a three-dimensional world for the kids and let them learn about what chemistry is. It's important not only if you want to be a researcher, if you are a talent in that regard. It's also important if you are working with concrete, or you're a carpenter, or you're a baker. It's all chemistry you're working with, actually. So chemistry is very important, and we should teach our young from the very beginning what chemistry is all about. And to recruit the talents, we should let them play. It's the playful mind that actually invents new things. It's necessary to be playful in order to come up with novelty. So research with no constraints is a very important issue, in my opinion, in order to get the best talents to choose chemistry, to choose physics, the two fundamental sciences that describes everything, and excuse me, including genetics and biology. So, I am from a family that was very privileged. This is my grandfather's farm, and this is the beauty of that farm, the meadows and the forest in the back, and here you see the forest. This is where my interest in chemistry really started. When I was a young kid there, I was watching this nature, this beauty, and I was thinking, how can that be? How is everything fitting so well together? And what is the reason that everything is adapting to each other here? The only reason I can really see is chemistry. So this is inherent in each atom that makes it up. So chemistry actually is a basic building block for making life. And that's why it's very important that we learn chemistry. So when I hire a postdoc or PhD, I'm of course looking at your factual knowledge if you're applying for a position with me. But more importantly, I'm looking if you have intuition. Do you have the ability to imagine stuff? Do you have this three-dimensional world in your head? Can you associate your knowledge so that you make new bridges about what you want to do? And so on. All of these creativity, collaboration, and most importantly, courage. Can you tell me that I'm stupid and that you want to do it your own way? That's a very good indication that you can become a postdoc in my lab. So factual knowledge is there, but it's way down the ladder. Because today it's very easy to get clever in a very short time due to the internet. But the road to success is never straight. It goes through a lot of problems, and most scientific projects actually fail initially. Also because if you want to have something that is important to do, you are challenging both yourself and your science project. So initially it will always fail, and then you can just look at different opportunities and determine your path. You fail and you rise again, and eventually you become very good at that. And you take the successes, and this is what you publish, this is what you become famous for. But this, the short thing is that you study all your life. It never ends. This is where I started my career. This is Carlsberg Laboratory. They were working with enzymes and biochemistry, so I was coming there to set up a synthetic lab. I got the help from the Danish National Research Foundation to set up a infrastructure for synthesis, and I chose to focus around combinatorial chemistry and taking all the organic reactions, putting them onto solid support, making organic molecules that could interact with biomolecules. So we made these kind of polymers that was compatible with organic chemistry and with enzyme assays and with recognition of proteins and so on. We could grow cells on these surfaces. And we then optimized all of these different reactions, perfect organic reactions that works 100% on our solid supports. So this is completely different from organic synthesis in solution, where you get this 50, 60, 70% if you're lucky. Here we could carry out 4 or 5 of these reactions quantitatively. During this work, we worked with the Diels-Alder reaction because we were trying to take a peptide hormone and put on a lipophilic side chain. You might recognize that as quite a valuable thing to do. We couldn't make that happen. It was almost impossible for us to react the alkynes that we would insert in the peptides with aceto-acid chlorides. And the reason was that instead of making the acyl group isolating the alkyne, we got the triazole. And this was a quantitative product in the reaction. Christian Tornø, who was my PhD student at the moment, he tried for months to make this Diels-Alder reaction work, and he threw out a lot of compound actually, which was very well made, was quantitative all the time, and this was a triazole that later became the click reaction. So we built a platform in this place. This is our combinatorial methods, and we can then screen in different bioassays on the solid supports. We can isolate compounds also in solution, and then we can inject them in mice and see whether here's inflammation, for example. We can grow cells on the surface of beads, detect molecules that interact with receptors that are expressed in these cells, and we can identify compounds using a microparticle matrix encoding, similar to a barcode. Just every bead contains this kind of barcode, which is randomly made in the bead. So we can make libraries of compounds of different structures here, or catalysts, and that is our technology platform that I made at the Kausik laboratory and took with me to Copenhagen University. So core to this is the polymers. Polymers are compatible with biology and chemistry. The other core to this is our technology for making combinatorial libraries. Here we can make 1 million compounds, 1 million different compounds in a week. So that's quite powerful. And then we can add an enzyme if we have some sort of indicator here. As you can see here, we can have different tags on these resins. If we have an indicator for enzyme activity, we can then identify the active compound or inhibitor. So what is click chemistry? Yeah, this is an alkyne. Many of you might know so, many of you might not. So I'll just call it a stack of electrons. It's the HOMO for the alkyne. It's full of electrons, so it's electronegative. And it's looking around for something electropositive. And for some unknown reason, copper 1 has a very high affinity for this guy. So copper 1 plus comes in and attaches itself to the electrons. It withdraws some electron density, and the proton sitting here at the end becomes acidic, and now the copper can take its place. This looks exactly the same as it did before, nothing different. But it's still a stack of electrons looking for electron density, electropositive ions. Here comes the second copper 1 ion. It peels off the electrons like that, just as a can of sardines. And inside is a positive charge. So it rearranges like that. And this is the empty orbital of the LUMO of the dimethyl complex here. And this LUMO, fortunately, fits almost exactly in the overlap with these electrons. So this is very electron-rich orbitals. This is electron-poor orbitals. So now we can just pour over the electrons and form the bonds. Looks like that, first with a metal. And first one metal goes out, and then the second metal goes out, and we have formed the triazole. Is that exciting? Not really. But what is exciting is that this reaction is completely orthogonal to all the other chemistries we know. So we can perform these reactions in the presence of completely unprotected proteins and DNA and carbohydrates. we can also perform reactions over in the carbohydrate protein space without touching these two functionalities. So this makes it completely orthogonal, so a completely different toolbox than the conventional organic chemistry toolbox. And this is what it looks like more scientifically. This is the scheme that I just showed you with all the steps. These are the crystal structures that prove that these steps actually exist. And here we have a situation where this is the catalysis we get with just copper without any assistance. And if we have a catalyst that has the right distance between two copper atoms, we get this kind of acceleration because this guy is binding here at these two, accelerating the reaction tremendously. It's so reactive that you should not mix it in neat because then you get explosions. I'm not good at standing still. So here are three guys. We can call them proteins or peptides. And this one here is an apoptosis activating cell guy. This one can locate some cancer tissue, and this one can enter the cell. Each and every one of those could be used for different purposes, but they are not very useful in killing a cancer cell. But if we now combine them by putting on the click residues, and we can do that very efficiently either by synthesis or by using the amber stop codon technology. And here we mix them in a container and steer them overnight. And if we mix them in the right ratio, we get almost one compound out clean, which looks like that. And now we can find the cancer cell, we can enter the cancer cell, and we can activate apoptosis. So we can build molecular robots using click chemistry, and this is a really powerful thing that I think will have a lot of applications in the future. There's actually 2 cancer drugs in clinic now that utilize this kind of click chemistry. So the only caveat with click is that it's very sensitive to oxygen. Initially it was thought that, okay, that doesn't really matter. So the barrier of oxidation to copper-2 is very low. It happens readily with oxygen present. So our synthesis will not continue because this is all moved over here if there's oxygen. So what about just adding a reductant like Sharpless did initially? Well, if you add a reductant here, you get your copper-1 back and the reaction goes, but you also get a large number of very reactive oxygen species. And these actually destroy your proteins, as shown down here, where you have the molecular weight plus 16, plus 32, plus 48. A lot of different compounds are formed in this case because of the oxygen present. So you have to get rid of the oxygen, and you have to take care to protect your copper-1 species. Then this is a real click reaction. So it's not really a click reaction because that is one of the criteria to have a good click reaction. But we still got the Nobel Prize. Here is a Japanese horseshoe crab. That is a very famous animal. It's 400 million years. Not these ones are not 400 million years, but the species is 400 million years. And it's like a pharmaceutical factory. It produces a beautiful blue blood, bright blue blood that is used by in medicine to discover whether we have bacteria in blood, because they have 2 macromolecules that encage the bacteria, and then it injects these toxins that kills the bacteria. So we were not so much interested in the interaction with bacteria, but more with the, can we use these drugs for making, drugs for humans? And the way they work, the way they kill bacteria is because it's a beta hairpin with 2 cross-links, so it's a very stable structure. And what we did was to replace these 2 labile disulfide bonds with triazoles, so we clicked them together. isolated this compound very purely. And then we analyzed by NMR, and we could see that they actually form a dimer in solution. So they are harmless when they are just together in solution, in the blood, in serum. But as soon as they meet a negatively charged membrane, something happens. So they come here and they get together, they make a pore. So they unfold and make a pore with the lipophilic parts against the membrane, and through this pore, all the interior of the cell is coming out, so the cell is perforated and it dies. So that's how these peptides work. And we then asked ourselves, is our peptide better or similar to the natural ones? We tried that with coli, Staphylococcus, Salmonella, and Bacillus subtilis. The result is that the natural one is active in the first 3 ones. In this one, it's completely inactive. But if we have our click bridge, it's active, almost as active in all 4 of them, including being active in Bacillus subtilis, meaning probably that this bacteria has the ability to reduce the disulfide bonds and remove the activity. So click can be good in establishing more global antimicrobial peptides. Another application of click chemistry I show here with the expression of proteases. This is inherently difficult because the proteases are proteases, so they are toxic to the host organism. They cleave the proteins that the host needs to survive. And therefore, you get a little bit of expression of the protease, and then everything dies away. So what can we do? Yeah, we can simply ask first, is it okay? And it was not okay for the coli. So what we do is we cleave a protein in 2 fragments, and we install the click residues in each fragment by amber stop codon technology. We express that separately in coli, and then we ask the coli if that's okay. And it was definitely okay because we got several hundred milligrams of each of these 2 fragments. So more scientifically, we made a small bridge, and we made a large bridge by amber stop codon technology. We inserted these in these constructs here. So these are the 2 parts for the short bridge. These are 2 parts for the long bridge. And you can see the expression here is very good. This is the mass spectrum of this material. In a linear mode and in ESI. And then we combined the 2 and clicked them together. And with a short bridge, we have some unclicked and some clicked. But with a long bridge, we can mix these 2 components 1-to-1 and click them together quantitatively to form only one product. And this product, we can then ask, is this product that we have made with these 2 proteins here, is it an active protease? Because we didn't know at that point. So first we click them together here and form the triazole in the center. And then we ask, can it cleave the substrate? And the fact is, it cleaves the substrate much better than the natural, not much better, but a little better than the natural protease, which is the lower curve. And this is the upper curve from fluorescence measured on this FRET substrate. So we can actually make proteases expressed in high yields in this way. So here we are in Stockholm, and this is the 6th of December, where we get informed about the whole entire celebration of the week in Stockholm. And then you deliver a small item, which is then maintained at the museum in Stockholm. So this is the first multiple column synthesizer in the world. And we deliver the paper describing it and this small item, and it's now up there in Stockholm. And then you get to sign a chair with a gold pen. So that was a very nice day. The next application is in structural integrity. So we would like to maintain structure, and the way that we can maintain structure using click reactions is by installing click bridges in proteins that fold well. We would like to achieve these by synthetic means, meaning that the natural proteins are much too large for doing that. So what we thought was natural proteins are much too large generally. Why is that? Why is a natural protein so large when the function is tiny somewhere on the surface of the protein? And the reason is in the economy of the expression of proteins. So proteins are tactic molecules, they are all amino acids, and that's why we have helices and sheets and so on. If we now expand the number of amino acids, including all the D-amino acids, we get no tacticity in our proteases or our proteins, and therefore we can fold them up like small bundles, much smaller proteins with folds that are very stable, and we can then further stabilize those by click chemistry. So that's what you see here. We take a completely random LD amino acid string and we fold it up. We optimize a hydrophobic core in the interior, install surface residues. This is all by modeling, what we call mutational molecular modeling, molecular dynamics. And then we install first the surface residues, and we find a good active site, install active site residues, and now we have an enzyme that is maybe 50 amino acids or 40 amino acids long. And we can synthesize all the varieties of these structural templates once they are made. So they are fully soluble at this small size of a protein. And here we see 4 serine proteases. We have made probably 20 of these structures. This one is the most active so far. It's almost as active as the most active serine proteases out there. It has a disulfide bridge and a click bridge here. But before we do that, we actually investigate the folding. Before we make the bridges, we investigate the folding in the dynamics, and you can see even very long dynamics give a completely stable fold without the bridges. Then we install the bridges, then we install the active site residues, and we get very active ligands. This is Mads Rosensaar, Aline, and David that has performed this work, and it's very exciting in our lab, I would say. So this is a protease, and this is the stability over time. You can see the distances really are maintained within this active site, and you can also see that we are below 4.5 in our MSD. So it's a very stable protein. We can synthesize this by solid phase synthesis in a very pure form. And if you look up here, you wouldn't say, what is that? Is that a protein? I don't know. It looks funny. There's a lot of things in there. And here we have the mass spectrum of the crude product showing that it's really a pure protein. We then make a library of substrates that are fluorescent with a fluorophore here close to the resin. If we cleave here, we'll get a fluorescent bead like that. We try that first with a natural protease subtilisin, and we get very active substrates out of that. You can see here we start getting rings that are increasing. The light is not really good for this, but you can see some very blue beads here, a few dark beads left after 24 hours. This is one of the more active proteases that are used for washing powder today. And here we have our protease. Starts out with completely black beads. We get some few here after 3 hours, so a little bit longer. Oh, sorry. A little bit longer than the subtilisin, but this actually is very selective. There's full of dark beads here, and you see only a couple of lighting beads here. Here's after 7 days, very clearly highly fluorescent. After 3 months, everything's fluorescent. So it clearly works as a protease. And we are currently looking into what are the specificities of these different proteases that we have made synthetically. I think it's a very, very exciting field because I find that this could be a new area of drug research. Can you make these kind of things synthetically so that they actually have the specificity for a pathogen? Then maybe you can use these ones in very tiny amounts as a drug instead of a large amount of some inhibitor of a protein-protein interaction. So this is what it looks like. The two bridges, the active site residues, the hydrophobic core, and all the hydrophilic environment making this protein. I'm extremely excited about this. So now we're back in Stockholm, and this is when you get the prize. So the king comes forward with the prize, and you take the hand, and he holds the prize. A little like that. You get to hold the prize for a while, but then you have to give it back and sit down, because that is because, like with me, it takes like 20, 30 years before you actually get the Nobel Prize, and sometimes some of the Nobel laureates, they have lost a little bit, and then they forget the prizes under the seat in the ceremonial hall. So you finally get it on the last day, and then it's your responsibility to bring it home. Yeah. Another application here. This is the antibody, and this is very large for the recognition of a small molecule. So we have here the variable regions, and they are matching the partner, so they can recognize with very high affinity actually antigen. But we would like to make small molecules like these that can actually recognize proteins, in this case an interleukin here. Can we make 3D proteins that work just like antibodies, 3 kD, instead of 150 kD? That would be really nice. So what we do is we make what we call a beta body. It has blue structural residues which also convey solubility in water on the one side of this one, and on the back side you have recognition residues that you can vary combinatorially. And we can use this in a design design method, because we know what the structure of this backbone is, and we can just vary these in the interaction with the protein. And here you see two of these interacting with each other. I think probably the first time small molecules really recognize each other in this fashion. We do a lot of calculations. In these calculations you use this force field calculation, but this part here is really a problem, because you are placing the charges on the nuclei of the atoms, whereas what we are really interested in is all these small dipoles between the surfaces that makes the interaction happen. So we are looking dynamically into this kind of interaction at the same time as we do the dynamics. And when we do that, we can make these molecules that really recognize proteins. So this is GFP, and a beta body that recognizes GFP. This is interleukin 1 beta, with a beta body that recognizes interleukin 1 beta. If we now put these two beads together in a solution, and then we add GFP at 60 nanomolar, you can see only the GFP bead is lighting up. This one here is not getting any light at all. Opposite, if we take the interleukin 1 beta with the Roche label, we don't touch the GFP bead, only the interleukin beta body. So here we have molecular recognition. So now we have processing and recognition. This is our future is to combine the processing and the recognition. But just to let me show you what happens when we click these together, here you have the interleukin 1 beta recognition, and this is when we have clicked it, it just strengthens the interaction tremendously. So these are the beta bodies that we are looking at today. We have made a sandwich assay in which we have one beta body on the bead, we have an interleukin protein, and we have another beta body with a fluorophore. If we mix the two beta bodies and the bead, nothing happens at all. If we add this 20 nanomolar interleukin 6, we get a highly fluorescent bead within 20 minutes. So the protein is coming into the bead and recruiting the other partner. So it works very well in a sandwich assay. The last example that I will allude to is this Alzheimer project, in which we are looking at two different things. We are looking at the Alzheimer peptides as autocatalytic self-processing materials, but also we are looking to see if we can dissolve Alzheimer fibrils that forms the basis of these plaques. There are many kinds of crystal structures, but the most predominant one is this one for a beta 1 to 42. It's folded up like that with two strands, making this kind of double strand. So we took that and thought, if we now take this cross section and make beta bodies that sit here on the surface, can we inhibit the fibrillation of the betas? So the first thing we did was synthesizing one of them, or designing and synthesizing one of these beta bodies sitting on one end, and you can see we can only decrease the rate of fibrillation by adding this one. But that's because we have it only on one end, so it can still grow on the other end, because these two ends are actually structurally different. So we decided we want to make them at both ends, and then we get a little bit of fibrillation, but then it dies away. So it actually works to solubilize these fibrils. And you can see if we make these two here, so this is Niklas Henrik Fischer and Line Andersen that has made these peptides. If we take two of them, we get no fibrillation, because in fibrillation it would look like that. Also, if we add these to already fibrillated peptides, so this is after 200,000 seconds, so a lot of fibrillation already happening, and then we add the peptide and we get to 0 again. So that's very nice, but we were not really satisfied. What you can do is you can take something that recruits the water and assists the solubility. So we, instead of taking threonines, we put in these amino acids, which are made from glucosamine. You can see this is glucosaminic acid. We protect it here with FMOC, and the hydroxyl groups, we get this building block that we can use in solid phase synthesis, and we install this at strategic points in the peptides, as you can see. It has the same configuration of D-threonine, so we can make these with D configuration of all the amino acids, which is advantageous because then they are not metabolically susceptible. We made 5 of these, shown here, and we interacted those with the fibrils. So this is fibrillation as it normally behaves. This is what happens if we add our peptides, and they are very strong inhibitors of fibrillation. So in particular, these two, peptide 3 and peptide 5, are very efficient, and what happens is that they form, as I said, these caps of water at the end of some oligomers of these peptides, take them into solution, and if you can imagine, if you do that at night and you go into your REM sleep, you can actually pump out all this debris during the night. So this could be a very good intervention for Alzheimer. And here you have the fibrils in the background and just all these different structures that we imagine is happening. So in a click summary, we have actually 4 click reactions. I've just presented this one today, but there's also the spark reaction. There's this one, the TCO reaction from 2008. And we have also introduced an intermolecular click reaction, which is this one. Very nice click reaction. And 3 of them are driven by delta H, and 1 of them by delta HS here. So they need an energy property in order to run as click reactions. So in the click reaction up here, you get aromaticity. Here you have the proximity and the ring formation. So with the Jungian chemistry, what we really want to do is teach them some of all this already before they start high school. And how do we do that? We start with 5 minutes in first grade every week, teaching them how hydrogen atoms comes together, how this can be used for energy production and so on. And then second grade, third grade, longer and longer videos that show them the 3-dimensional work of the chemistry. So you go into the DNA molecules, you look at what the strings, how the strings are behaving and so on. At the later stage, in the beginning, it's just two atoms that comes together, share the electrons, where is the electron density in this molecule, and so on. That will give them a very firm understanding of chemistry before they start with molarities or how things work, kinetically. They will understand the kinetic scenario completely by just seeing how small molecules move around fast and large are tumbling around like that. So we call this institute with artificial intelligence that helps you find the right thing for your grade in your week, and then you get this sent out. This is very, very nice because this can very easily be made global by taking off the sound and putting on Swahili or Portuguese or whatever, and then just ship it there. So it's possible to do this for the entire world at the same time, and we are trying to establish an institute for chemistry animation. So here we are back in Stockholm, and this is my wife here in the first row. She's dressed up very beautifully because she's very proactive, and when she saw this dress at the exhibition at the museum, she said, "This is a dress I'm going to wear at the ceremony." So she called the Arts Institute in Stockholm, and they in 24 hours made this dress because, of course, it didn't fit the one in the museum. So they fitted her within 24 hours with this dress. So she's looking very beautiful, but that's a danger because this here is the Prince Philip from Sweden. He stole her off my arm and took her to sit between him and Prince Daniel. I was sitting down there. But that was okay because the day after we went to a very intimate dinner at the Royal Palace, 130 guests, and we were then invited to sit together with somebody at the table, and I was very lucky there. This is Victoria, an absolutely wonderful woman who is taking over the responsibility for Sweden in a few years. So with that, I would like to thank all the people who have been involved in the click chemistry. In particular, I have to thank Christian Wenzel Tornø, my PhD student at the time. But all of these people has been involved in the development of this technology. I would also like to thank my supervisor and Michael Boltz from Copenhagen University as the leaders that have accepted my scientific talent. And then I would like to thank my family and Phaedra, not the least, for accepting my late hours at work, because this doesn't come for free. Thank you very much.
Thank you for the inspiring talk. And I think we loved the bits in between as well, where you kind of showed the journey along. I'm sure there are a lot of questions. Probably I'll open with one question. You showed that at some point in the recognition process, the dipole were very important for the interaction of the peptide, and point charges, which are usually described in the force fields, were not enough. So what was the cost which actually made it highly specific? Was it?
No, we are actually not having this incorporated in the algorithm. We are actually using our visual interaction. We are constantly interacting with the programs. I'm not a believer of programs that can do everything for you. I think they are like a calculator in the old days. They just calculate what you put into them. And you need to be interactive with your computing. And we are that during this mutational molecular dynamics, where we're actually changing things continuously and then doing small molecular dynamics bits and pieces until we funnel into the combinatorial space of the molecule and end up with something that is really nice in interaction.
Very interesting. questions from the students. We have an exclusive interaction of faculties with him, so I'll just ask that if students have the question, this might be very interesting. Yeah, Abdul.
Very nice talk. It was very intriguing. But as you said that you have developed beta bodies nearly around the size 3 kDa, right? So, and that was very able to solubilize fibrillation in Alzheimer's disease as a model. So I was wondering that will it be, being the smaller size, will it be able to cross the blood-brain barrier? Have you looked into it?
I think we need assistant molecules, and we are working on penetrating peptides at the moment, trying to make artificial small peptides, again with click bridges and stuff like that, long fatty chains and so on, naphthalene residues in the side chains to interact with the membranes. And they are very efficient in transporting across the membrane. We don't know if they can come across the blood-brain barrier yet. That's a little longer story, I think.
And what would you like to tell about that? Does it actually mount an immunological response if you'll,
No, we have tried this out in mice. So the D amino acids do not bind to any of the immune molecules. So we have looked at all the cytokines and basically the weight of the mice before and after, and the TNF alpha and so on. And there's none at all of these indicators show any immune response.
Thank you.
Yeah, over to Devu.
It's an excellent talk. Actually, part of the question was already asked. I'm a researcher from IGIB. I wanted to know about clickable moieties in nucleic acids, and is there any kind of an immunological reaction to that? If you have any idea?
I don't know about the DNA and immune response at all, so I cannot tell you. We are very much protein focused, but I know that people are using clicking DNA and peptides together, so I don't know if that is used for any human use or whether it's just biochemical studies. I cannot really answer you very well there.
Thank you.
Yeah, Dr. Abhay.
So is it possible to make click mimetics of conventional drugs and
Of what?
click mimetics of using this click chemistry, the molecules which is similar to the conventionally made small molecule drugs?
Yes, so I know 2 drugs that are available, no, not available, they are clinic today, which are made in water by coupling sugars together with aromatic residues in click. So in a few reactions, not like in a 10-step reaction anymore, few reactions in water, and you get active compounds that are very efficient in fibrosis in the liver and the lungs. But I think that you will see many, many applications of this in the near future. The problem is that it takes 10, 20 years before you get through the entire process and can release your drug in the market.
Okay, one more question.
So when are you synthesizing that P bodies? So why their intramolecular click is happening, not intermolecular click, like two?
Okay. Yeah. So what we do there is for these particular ones, if you make large cycles, you have to cycle them in solution. Small cycles you can make on solid support. Large cycles you have to make them in solution. Otherwise, they will cyclize around the PEG chains in the polymer, and you never see a molecule. So we take them out of the resin, and then we make a very dilute solution of it, and we inject it into the click mixture. So every time a molecule hits the click mixture, it will click, and then it's done. And it's really selective. We can get it like 100% to that.
Yeah,
But you're right in the sense that if you don't do that, if you just mix it.
Cell wall reaction, and then it proceeds to the cell for the apoptosis.
So what happens is when it hits a negatively charged membrane, it actually opens up, and that it migrates into the membrane, and then it opens up completely and makes a pore.
For the cell wall part, for the bacterial cell wall that is there, so is there a specific reaction that is involved there so that it is able to cross?
No, it's not specific in the crossing, it's specific in the interaction, because the negatively charged cell membrane recruits these very efficiently compared to the human cell membrane, which is essentially neutral.
Sure, thank you so much.
Okay. We can also move to some non-scientific question.
Absolutely.
Which may inspire the audience here. I read from your Nobel Prize biography page that not all was very full of positive funding back when you were at the associate professor stage. So we are, for example, you said that in 2005 when you were in University of Karlsruhe, there was a complete shutdown of chemistry lab, and apparently the whole shift went from chemistry to beer.
Yeah.
So at that time, you kind of also shifted your research, and then later also moved to another university. So at the time where we are constantly chasing funding as scientists. What would be your advice during this turbulent times?
That situation was quite special because Carlsberg statutes actually mandated that there had to be a fundamental research laboratory, and the spillovers from that would be used in the beer production. And they translated that into all the research has to be for beer production. So that was very difficult to handle when you are working with fundamental sciences like I was. Even though I was the last one that left the sinking ship. So all the other professors, they diffused out very quickly. I was the last one to leave the ship, and I did that in 2011. So I tried to follow up and try to do some fundamental things with what they wanted to do with the materials for bottles and so on. But I think that you should follow your heart. I think it is important to make the decisions early. As they say in drug industry, it's better to fail early than to fail late. And I think that's the same for your research. If you fail, if you don't get what you want to get, then ask yourself what you want and go there. Find your mentors, as my wife always says. Find your mentors and your advisory board for your personal, growing up. And they can advise you what to do, because it's very difficult as a person yourself to make those decisions. And then get the help you need to get where you want to go.
And also we have a lot of students in the audience, and you also mentioned that at some point you had to choose your career between physics, chemistry and computational sciences. I mean, we know from your lecture now that everything is chemistry and that you have reached a stage where you look from that lens. But at that point, when you had to make the decision, what were the factors that you thought about?
At that time, computational methods, computational chemistry in particular, was extremely new. This is a long time ago, as you can see on my right here. So there was not really a lot of computers around. Actually, our first computer programs were punch cards. So we had this big box with 1,000 cards, and we put them in the machine. And while it was reading, it was running the program. So that's how you ran a program in the old days. But I found that very fascinating that you could actually do this with, we made a molecular simulation program already then with Fortran. It was really fun. So I have maintained that interest for programming all my life, and I have made a lot of different programs for the laboratory, controlling instrument or analyzing images or all sorts of things that we do in the lab, mass spectrometry and so on. So programming is close to my heart, but chemistry is everything.
Yeah, I think we are towards the end. I'll just also say that towards the end of your career, you have moved into entrepreneurship.
Yes.
And that means that there is sufficient scope of taking click chemistry to applications.
Yes.
And so what would be, if you could elaborate a little bit of your efforts in that direction for the young students?
Yeah, so whenever you have something that looks good and could be solving problems out in society, immediately go to your tech trans unit in your university and disclose this. It doesn't cost you anything to do that. And then the tech trans unit will negotiate with you, what can we do with this? Is that possible to patent and so on? And after you give the advice, they will actually probably do most of the work for you because they're also very interested in getting these patents. Just make sure that you set a time limit on the time they can use before you can publish your paper. So that's a very important thing to remember. Yes?
One more question.
Thank you so much for the talk. My question is related to problem like which I'm thinking that you might encounter while doing your entire research. Like, did you ever face any challenges while doing it from the quantum perspective? Like, were you looking chemistry in a very classical manner, or did you face some challenges? Then, if you did, then what challenges were there, and how did you solve that, whether using quantum or classical mechanics?
I'm not a quantum chemist, so I have never used any quantum methods or anything like that.
Entire chemistry?
I think quantum chemistry is extremely interesting, and I think a lot of solutions to the problems that we still don't have solutions to, which are many, including molecular recognition. That's why we cannot design anything today, really, very easily. Okay. But if we really understood molecular recognition from a quantum level, I'm sure we'll get much better results. So there's a lot to do with quantum chemistry, I'm sure, but I'm not the one to do it if you look at my gray hair.
Okay, great. I think with that, I'd like to thank you for an excellent lecture, and we have a special memento for you, and I'll call upon stage Dr. Chetna, Ajay, and our director to be present on the stage, please.
So we would like to thank you with some things that you remember about India. So Ajay, if you can step over to the stage. So we have a tradition of giving a book to the visitors who come to IGIB. So we have chosen a book that details the natural history of India. It's a beautifully written book and with a lot of beautiful pictures. So hopefully you'll be able to read it on your journey back. So another thing that we have for you to take home is painting. So India is home to around more than 700 tribes. So this is, and with each of them having a very distinct art form. So the painting that we are giving to you is from the middle of India. It's the Gond tribe painting. And it shows a peacock, which is our national bird. So in a very distinctive style. We'll take a picture of you with it.
Thank you.
We would also like to invite Dr. St. Clair to the stage. We have a gift for you to take home to your cold.
Thank you.
I guess this is really warm for you, right? Here. So this is a shawl, also from a tribal art form, a special weave from the north of India. Thank you so much, both of you, for visiting.
That would be very good in Denmark.
Thank you.
So much.
Yes, thank you very much. Thank you. What's the next thing? Sit down.
Yeah. I just want to take 2 minutes, please. Please settle down. I want to just conclude the day today by a vote of thanks. We are extremely thankful to Indian Council for Cultural Relations under Ministry of External Affairs for arranging this talk through CSIR headquarters, ISTAT department. I am also thankful to internal IGIB colleagues, technical staff, my lab, who have helped me in organization of the lecture. And to just seal the whole evening, I would request, we'll have photo session in 2 rounds. First, I will request all the faculties of IGIB to please come on stage. And also if DU teachers, Delhi University teachers can join us. So we'll have 2 photo sessions, one